This figure is showing the technique used in cloning to produce, for example, Dolly, which is probably the most renowned clone that people are aware of. The thing that made Dolly so fascinating was embryo splitting sort of makes sense. In other words, if the two-cell or four-cell stage is going to ultimately develop into an entire offspring anyway, then it really doesn't seem that surprising that if we split the cells at that stage, that each cell could ultimately develop into a complete organism. But the question that scientists had was, after cells differentiate and specialize, do they still have the ability to basically revert themselves and ultimately end up thinking that they're a fertilized egg and that they start the process of dividing all over again? So that's what Ian Wilmot's work was with Dolly. This was the first attempt at what was called, or the first successful attempt at what was called somatic cloning. So the cell from which Dolly ultimately derived, or the nucleus from the cell from which Dolly ultimately derived, came from an udder cell, or from the mammary gland. In other words, this particular cell had specialized to produce milk. That was its function. And so what Wilmot and his colleagues did was they took a donor egg and they removed the genetic material from that egg. Then they took the nucleus out of the mammary cell and they injected that into the egg, and then they exposed this situation to an electrical shock. And what happened was the electrical shock causes changes in the permeability of the plasma membrane that surrounds the egg. So what happens is that nucleus from the donor cell is able to slip in, you could think of, into the egg. Now, at this point, the egg believes that it's been fertilized and that it's now a diploid cell. Because remember, we took the nucleus from a non-reproductive cell. So what happens is you take females that have been synchronized, in other words, falsely led to believe through hormone manipulation that they're pregnant, and you put that embryo into the female. And that female, if the embryo implants successfully, that offspring will develop into a clone. So in other words, a sheep gave birth to Dolly, who was a clone of another sheep. And again, that process is somatic cell cloning, where you take the nucleus from an already developed cell and basically manipulate it to develop a clone. Now, the thing that was so interesting, or another thing that was so interesting about this, was again that it showed that all of the DNA required to make a complete new organism was found inside of a cell that had specialized to produce milk. So this just reminds us that when cells specialize, they don't lose DNA. What happens is portions of the DNA become used in that cell to accomplish the particular function that that cell is carrying out. So it's a really fascinating situation.